Hello, I am Wassilios Meissner. I'm professor of neurology at the University in Bordeaux. I'm also co-chair of the French Reference Center for MSA. I will talk today about new MSA therapies in the pipeline. Most will deal with alpha-synuclein, which is the hallmark feature of MSA as illustrated here. You can see here an image of some brain tissue of a patient who made brain donation. And you can see here in red the alpha-synuclein accumulating in cells we call oligonucleotides. Alpha-synuclein also accumulates in the brains of patients with Parkinson's disease, but in contrast to MSA, most of this occurs in neurons. This is here the image of the illustration of the protein, which has physiological functions, and one of these functions is to be involved in the release of dopamine, as illustrated here in red. Dopamine is important in motor control, which allows us to perform a voluntary movement. And what happens in MSA and Parkinson's disease, that the protein here, which has physiological functions, then changes its conformations and accumulates in either these glial cells or in neurons in Parkinson's disease. We know more and more about the mechanisms underlying this misfolding of the protein of alpha-synuclein, and the different treatments that are currently in development try to target in a different way the accumulation of alpha-synuclein. But before I share with you the ongoing trials and recent trials, mostly targeting alpha-synuclein, I would just like to share with you the different ways how to tackle alpha-synuclein. And there are 4 different options currently. One is to decrease the genetic expression, which is probably not the most straightforward in MSA, because MSA is not considered as a genetic disorder, but there are some rare disorders where there is genetic overexpression of alpha-synuclein. Clearance is another way, and you should keep in mind that usually proteins have a cycle where there is some synthesis, there's a use, for example, for the release of dopamine, and then there is some metabolism, so digestions of these proteins, which are then reassembled into amino acids, which can then be used to reassemble again proteins. And there are a few attempts currently in development that try to really enhance the clearance of the protein, and by that means to reduce the accumulation of alpha-synuclein. Another way is to interfere with the aggregation, so with a process that forms 2, 3, 10, hundreds, thousands of these different molecules. And there are some small molecules that can interfere with this process, which one also being very close to a clinical trial in MSA. And the most advanced is immunotherapy. There's either active or passive immunization. Active immunization means the injection of a vaccine, which then induces an immune response against alpha-synuclein, while passive immunization means that the antibody targeting alpha-synuclein is directly administered into the blood. But before we come to the different trials, just a very short focus about clinical trials, or the development of clinical trials in general. There are 4 phases. Phase 1 is the earliest, with focus on safety and tolerability. It's usually the first administration in humans and mostly involves healthy volunteers. Some patients may also be part of these trials, but usually it's healthy volunteers. And once these trials have shown that the compound is safe and well tolerated, then it moves to phase 2, which again, the main focus is on safety and tolerability, but then already there will also be some kind of testing of different doses to prepare the phase 3, which is the phase where the efficacy is really evaluated in a large number of patients. In a rare disorder, usually a few hundreds of patients, and in larger diseases, this number may go up into several thousands of patients. Still, at that stage, safety and tolerability is also monitored in a larger number of patients, and based on the efficacy of these phase 3 trials, then market approval is usually obtained. And after this market approval, still the monitoring of long-term effects, safety, tolerability, and efficacy is ongoing. So these are the different steps for the development, and each step takes usually several years, which gives you also an illustration how long it takes from a preclinical validation, for example, in cell culture or in rodent models, to a market approval. What is also important to understand is that in these different phases, you can see these are quite historical data, but still it very nicely illustrates the bottom line that in phase 1 and phase 2, there's really an increasing pipeline in different disorders. But then the challenge is really that the compounds prove in these 1 and 2 phases to be safe, well tolerated, but also to have some potential efficacy to move these then forward in phase 3. So when it comes to alpha-synuclein, this is here a summary of the current pipeline. On the left, this is AFI2, PD-L1A, PD-L3A, which is active immunization. I will come to this in a minute because we had the chance to conduct a phase 1 trial in MSA patients in France. There are 2 compounds, passive immunization, 2 antibodies that have already moved to phase 2, and the other one is in preparation and will very shortly start enrolling patients with MSA. There's 1 compound targeting genetic expression, which is also ongoing. I have 1 slide to share with you about this trial. 138B is a compound that interferes with the aggregation. The phase 2 in MSA is in preparation. There has been a phase 1 study in Parkinson's disease, which showed that the compound was well tolerated. And then there was recently a phase 2 with sirolimus, which is the compound that enhances the clearance I mentioned in one of the previous slides, the clearance of alpha-synuclein. This trial, unfortunately, was negative and was stopped prematurely in April 2021. So when it comes to active immunization, this is a study we had the chance to conduct within the framework of the SYMPATH consortium. The trial was conducted in France, 2 sites of the French Reference Center for MSA in Bordeaux and Toulouse. We enrolled 30 patients who were assigned to either receive PD-L1A, PD-L3A, or placebo, and then patients were followed over a year. The study visits were mainly devoted to safety because that was a phase 1 trial. There was a phase 1 trial. We did also MRI and lumbar puncture at baseline and then later during the study, again, mainly for safety purposes. But the data we could gather out from the MRI and the lumbar puncture will also help us to understand how biomarkers progress over time, and this provides important information for the design of future clinical trials. So the main question, because over the year there was no relevant issue with safety or tolerability except for local skin reactions because of the vaccine, which is quite classical in vaccines. But then the question was, were these PD-L1 and PD-L3A able to induce antibodies against the peptide, the immunizing peptide? And as you can see here, in blue, this is the placebo group, and in green, this is the PD-L1 group. So patients received 4 immunization, which were just every 4 weeks over 12 weeks. And you can see here an progressive increase of the titers, which then reduced over time. And then the patients received boost immunization after 36 weeks, and that was able to boost again the titers. When it comes to PD-L3, as you can see here, there was also an immune reaction, which was really much, much smaller than with PD-L1. And the additional question, because this is here the titer against the immunizing peptide, the question, of course, the major question is to what extent this immunization was able to induce antibodies against alpha-synuclein. As you can see here, PD-L1 was able to induce antibodies against alpha-synuclein again in a progressive way, some reduction then over time, and the boost was able to again enhance the titers, while PD-L3 was really unable to induce any relevant titers against alpha-synuclein, which means, as a bottom line, that it is of interest to pursue the development of PD-L1, but PD-L3, since there was really no generation of antibodies against alpha-synuclein, would not be reasonable to pursue in future trials. The Horizon trial is a trial that assesses the impact of what we call an antisense oligonucleotide. So these are compounds that interfere with the genetic expression of alpha-synuclein, as I mentioned already. There's currently a small multi-center phase 1 trial, so very early on, patients receive the administration of the compound, which needs to be administered through lumbar puncture in over several weeks. It's 40 patients. And since this is a phase 1 trial, the primary outcome is safety and tolerability. But of course, this trial will also be important to understand to what extent this compound can interfere with the expression of alpha-synuclein, reduce the expression of alpha-synuclein, and if that is then potentially of interest to test, to challenge this compound in a larger trial in the future. Another approach, a little bit different, because the main focus is iron, but we know that iron, there's an interaction between iron accumulation and alpha-synuclein and oxidative stress. So there is iron can interact with the disease process, with the mechanisms, by stimulating the action of alpha-synuclein. And there's one trial that has started enrolling patients. First patients were enrolled in New Zealand and Australia. This is a trial that will also involve centers in the United States and in Europe with ATH 434, which aims to interfere with the iron accumulation. And just to give you an illustration, this is an MRI of a patient with MSA, and you can see here these black parts are iron, is iron accumulation in a region we call the putamen. This is a region where dopamine is usually very high, dopamine concentrations are very high, and within the disease process of MSA, these dopamine levels are reduced because of the loss of the dopamine terminals in that part. And in parallel, there's the accumulation of the iron as illustrated here. So this is a trial, phase 2 trial in 60 patients, so a rather small trial. And the main objective will be to see to what extent this compound can interfere with the accumulation of iron in the brains of patients who are part of this study in preparation then of a larger study. Changing completely the focus. So it's not only, everything is not only about alpha-synuclein. This is a trial that is currently led by our colleagues, by Tom Fultini and his group at UCL in London, with an approved antidiabetic drug called exenatide. There has been a trial in Parkinson's disease. We have done some preclinical work in MSA transgenic mice and also on post-mortem brain tissue of MSA patients, where we could see that insulin signaling in the brain tissue of the patients and also the mouse model is impaired. And when then challenging these transgenic mice with exenatide, we could see that there was an interference, a positive effect on the cell loss of the dopamine neuron cell loss. And that motivated our colleagues in London to initiate that small trial in 50 patients, 48 weeks. And here, the primary endpoint is the difference in the UMSS score. UMSS is a scale that allows to assess the severity of symptoms, slowness, rigidity, tremor, gait, speech, and also activities of daily living. And that gives them a total score. And this scores, we know that these scores are progressing usually over time. And the idea is, or the question is, to what extent that drug can interfere with the progression of the UMSS score over time. So this is ongoing small trial, and if there's a signal, then probably it would be important to validate this approach in a larger randomized double-blind trial. Another approach which is currently ongoing, led by the colleagues at the Mayo Clinic in Rochester, is to evaluate the efficacy and safety of autologous mesenchymal stem cells. There has been a study a few years ago from Korea, where in a small number of patients, was a small trial in patients with the cerebellar phenotype subtype of MSA, where there was a positive effect on the progression of the UMSS, the scale I've just mentioned in the previous slide. And based on that, the colleagues at Mayo have started to develop a program. There's a first phase 1 and 2 study that was published in 2019. And this is just here the summary of that trial. It required also the administration of these stem cells via lumbar puncture. The primary endpoint in this was an early try where safety and tolerability after 12 months for patients were followed at least for 12 months. And then, of course, there were also some secondary exploratory endpoints to understand if there's potentially an impact on the progression of symptoms and also on factors, mainly neurotrophic factors in the cerebrospinal fluid that may help to understand how that therapy could work in patients. 24 patients were randomized, small study. It was an open label, so the doctor and the patient knew that they were receiving the treatment, which is also important when it comes to a potential placebo effect. There was no serious adverse event. There were 3 different doses, and in the highest dose, there was a dose-dependent thickening of what we call the chordae equina. As you can see here, this is an imaging of the spine in one of the patients who's enrolled in this trial. And in patients with the highest dose, there was a dose-dependent thickening of chordae equina nerve roots, which then led to transient low back and leg pain in some of these patients. There was also some, as I mentioned, exploratory investigations. The UMSIS progression was slower compared to a historical placebo group of a trial that the same team has run a few years ago, but there was no real comparison, and it was an open label design. So we should be very careful in the interpretation on efficacy measures in that trial. But that finally led the colleagues at Mayo to implement a larger trial, which is currently recruiting 76 patients, double-blind, and here really there's a comparison between the stem cells and placebo, and this trial will probably help us to understand about the efficacy of that approach. And finally, before I will close, just a short word about the MSTAR trial. That was a large phase 3 trial that was conducted over the last few years with verdiperstat, which is a compound interfering with neuroinflammation. And as you can see, that was really a large phase 3 trial, 336 patients over 48 weeks. And the primary endpoint was the change from baseline in the UMSIS score. So here really efficacy phase 3 trial, that was really a phase 3 efficacy trial compared to placebo. And unfortunately, the top line results that were reported in September 2021 showed that the study failed its primary and secondary endpoints, and the study has recently been stopped. So we are still waiting for the publication of the full results, but the results we announced so far show that this approach unfortunately was not efficacious. So to summarize, there's a growing list of compounds with beneficial effects in preclinical MSA models. I haven't discussed this, but please trust me, there's really a lot ongoing, many studies testing compounds in preclinical models of MSA. Active immunization with PD-L1A, PD-L3A was safe and well tolerated in a phase 1 trial. 2 trials assessing an ASO and passive immunization are currently ongoing, and more compounds targeting alpha-synuclein are expected to enter soon clinical development. Beyond those approaches specifically targeting alpha-synuclein, the additional compounds in clinical development, I mentioned the approved antidiabetic drug exenatide, ATH434, targeting iron and indirectly alpha-synuclein, and then the mesenchymal stem cell approach currently developed by the colleagues at the Mayo Clinic. Thank you very much for your attention.